So hi everybody, my name is Yun, and I'm now a third-year PhD student in the University of Florida, and under the supervision of Dr. Mingzhou Ding. And I'll be happy to introduce our work in imaging trigeminal neuralgia pain and insights from combining AI and Granger Causality. So first, I will introduce our experimental paradigm first. We are doing the simultaneous EEG fMRI recordings. And our pipeline is divided into 4 parts. The first part is for the resting period. We'll take the 7 minutes continuously. And after that, we'll do the pain tracking period for continuous 20 minutes. And in these 20 minutes, the patients would have a tracking ball to track their current pain scores. It's from the range of 0 to 100. And afterwards, we'll track the T1 images for 5 minutes. And finally, we'll track the DTI images for 17 minutes. Between each of the sessions, we'll have 1 minute pure resting period. And you can see the figure below. It is a sample pain level fluctuations from one of our sample patients. We can see some of the fluctuations in the tracking pain curve, which demonstrate that the patient is experiencing high pain or low pain or moderate pain. So after that, we come to our aim 1. Besides common machine learning algorithms, we would like to try to use the deep learning to help us identify some of the signature centers of TM pain. So just as in figure A, this is a really simple version of our framework. We'll input fMRI images, and after that, the images will be fed into a RANS next ready with non-local block structure, and we modify the final fully connected layer. So for this, we can not only do the pain level classification, we can also do the pain score prediction, which we have 2 trends. One is for the score regression, and another is for the classification. And finally, after we have well-trained our pipeline, we'll use the GradCam to help us generate the heat map from all the patients. So figure B demonstrates one of our results in the pain score regression. We can have a R value of 0.838. We're using the predicted pain scores versus the real pain scores by the patients. So that demonstrates that the deep neural network can definitely help us find out some of the initial data distributions in the data. So the figure C demonstrates the heat maps from our results using the GradCam. We can see the insula, the ACC thalamus, which is really common pain signature pain centers showing up. And also we find the S1, S2 visual cortex, the medial prefrontal cortex, as well as the chordae and precuneus also showing up. And these regions are also shown as pain-related regions in some of the recent literatures. So that means our DNA results match with some of the recent journals showing the pain matrix. So which makes our DNA results more, it is more understandable and more maybe acceptable by most people. And after that, we'll come to our aim 2. We would like to find out whether there are some kind of the causal interactions between some of the regions which can predict the chronic TM pain. And we have pre-chosen some of the pain-related the regions, including the dorsal ACC, thalamus, and S1 insula and chordae. But we only show that the results between the dorsal ACC and thalamus here. And we found that the GC value from the thalamus to DACC, but not the DACC to thalamus, is calculated over the entire fMRI scan session, the 20 minutes pain tracking session, can help us predict the daily experienced pain levels, which means that the information flow between the from thalamus to DACC can definitely showing some of the useful informations. So that may provide some of the more innovations to our future study. So this is a really simpler version of our work. And thank you so much and I'm happy to answer any questions if any. Yeah, thank you.
Thank you very much. Excellent. So that was a lovely distillation of your findings and a really nice experiment. You also left us 30 seconds. Now, there don't seem to be any questions written down. And since I'm fielding the questions, what I suggest is that people, if they have any, they write them down for you and you could maybe stay and answer them online. One thing I did want to ask you very briefly, you kept a sort of real-time measure of the level of pain. How did participants indicate their pain level as you went through the scanning? I think I missed that.
So I'm suggesting you're referring to the tracking ball.
Yeah. Okay.
Yeah, the tracking ball is the pain scores is in the range of 0 to 100. And in the 20-minute session, the patients would just move the tracking ball, and that demonstrates their current pain scores in the current period.
Thank you. That's very clear. Thanks for an excellent presentation. That was really good.